Hello everyone, good afternoon. Good afternoon everyone. Yes, hello. Okay. So as you know, time is already up. So I'll request everyone, if you are familiar with any other friend of yours, please tell them or inform them that the session has already started. Okay. So please inform your friends so that they can also join. Okay. Yeah. Right. So, hello everyone. As all of you know, my name is Ghazala Firdaus and I'm one of the biology faculty in our campus. Okay. So today we are going to do the question paper discussion, which has been given to you either Saturday or Sunday. Okay. In your Navodaya, we have given you the full test syllabus. Okay, full syllabus test. So from that question paper, I will be picking some of the questions which might be felt difficult for you or it might have the critical thinking type of questions. So that type of questions I have taken and we are going to discuss those questions in this session. Okay, so before starting our today's session, I'm going to launch the technical poll. Just let me know if you have any internet issues. If I have any internet issues, कुछ तो receptivity का problem है नहीं है ठीक है. So इसका हम लोगों को पता लगना है. So for that I'm going to launch the technical poll. Please let me know if you have any problem. Okay. So I have launched the technical poll. Please reply to the questions. Am I audible? Is the screen visible? Am visible or not? Okay, so these three questions you have to answer. Yeah. Okay, so most of you say that I'm audible and I'm visible. Great, great. Then let's move ahead. Okay. So then, without any further delay, let's start today's session. So our first question of the paper is, the question is, crossing AABB and small ABB, the probability of capital A small b, capital B small b would be in F2 generation. Okay, options are 1 by 16, 2 by 16, 8 by 16, 4 by 16. Okay, so these are the options. And what you have to do, you have to know the probability of getting this combinational genotype. Okay, you have to search whether you are going to get this combination or not. Yeah. So in the F2 generation. So for that, what are we going to do? Let's see that in the next slide. So here, what are we going to do is very simple. First, the question was asking capital A, capital A, capital B, when we cross with small a, small a, small b, small b. So this is the parental generation. Okay, ये हो गया हमारा parent. Okay, so this parent will be given of the gametes and will get the F1 generation. Okay, we will get the F1 generation. So what do we get in the F1 generation? That is very simple. Okay, if we cross it, we are going to get capital A small a, capital B small b. So this is what we get in F1 generation. So after this, what we are going to do? We are going to do the selfing. Okay, we are going to do the selfing of this. So selfing में क्या हो जाएगा? Capital A small a, capital B small b crossed with capital A small a, capital B small b. So for this we have to take out the gametes, right? So for this we have to take out the gametes. So let us take out the gametes here. So gametes are very simple. I have told you how you have to take the gametes. Okay, so like this. Ye, okay, so this with 1 and 2. Then again this with 1 and 2. Okay, so capital A, capital B, then capital A, small b, then small a, capital B, then small a, small b. Okay, so these are the gametes, and again here also same gametes we are going to get. So that I am writing here. So after this gametes, if we are doing the cross here, ठीक है, अब हम यहाँ पे cross अगर करेंगे, so we'll consider among this one as male and another one as female. So these are all the gametes that you see here. These are the gametes. So we are crossing between the gametes. Okay. So now, what actually you have to do is you have to, okay, see the probability of this combination. Okay, so ये जो यहाँ पे combination है ना, इस combination का probability आपको देखना है F2 generation. So now we are going to see in the F2 generation. Okay, so I will write here in the F2 generation, that is this. Okay, ये है आपका F2 generation. So let us count here. Let us count here. Okay, what are we expecting? I will write here. Expected or probability. Okay, or probability. So what is the probability? We are checking the probability of capital A, small a, capital B, small a. Now we have to search for it. So let's start searching. So 1, 2, 3, and 4. Okay. So what we are getting here, we have total 16. Okay, we have total 60 offsprings. So in this 60 offsprings, so our probability, okay, so the number that we are seeing is, you can see here 4. Okay, हमको कितने मिल गए? We got the 4 numbers. So this is what we were looking for. So capital A, small b, capital B, small b is 4. So for this, what would be the answer? We can simply write. So answer for this question is 4 by 16. Okay, 4 is because there are 4 probability offsprings. Among how many offsprings? 16 offsprings. So this is the answer. Now, where is 4 by 16? Yeah. So answer D. Okay. So answer for this question is 4. That is D. Is everybody clear with this? Does anybody have any doubt? No? Then we'll move on. Okay, then we will move on. Chalo. Now let us see the second question. This is also one of the important questions. See, these questions are taken from the very important chapter, principle of inheritance. तो यहाँ से जो क्वेश्चंस आते हैं ना, एक तो पेडिग्री के क्वेश्चंस आते हैं, एक प्रोबेबिलिटी के क्वेश्चंस आते हैं, okay, and then there will be the Mendelian disorders and the non-Mendelian inheritance. So इन सब से questions आते हैं बेटा, numerical questions, and we have to be ready and we should be knowing how to solve these questions. Okay, so here in this question, what they say is that a family has 5 girls and no son. The probability of the occurrence of son in 6th child is. Okay, so for this, what we have to do? See, there is a family. Okay, as you can say, there is a family of mother. Okay, family of mother and father. So these people have how many child? Okay, they have 5 girls and they are expecting 1 more. Okay, so that expected one, okay, they are expecting 1 more. Okay, so what the question says, family has 5 girls and no son. So what is the probability of son? So here what we have to search is, your question is the probability of getting son as their child. Okay, probability of son in the generation. Okay, ये हमारा काम है, and we have to find the probability of the son. So for this, what we have to do, we simply have to plot a simple kind of this. Okay, so what happens in the father and there is a mother. So what type of chromosomes we have? So for the father, we know that he will contain X, Y. Okay, and the mother will contain XX. Okay, if we take out the gametes, what the gametes will be? XY. Okay, then XX. So there is a small cross between them. Now what we are going to do, we are going to look after the cross. That is this, 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 this. Okay, so the first one is, excuse me, I'm really sorry. Yeah, so the first one is XX. The second one is also XX. Okay, the third one is XY and the fourth one is again XY. So what we are seeing here, so the probability of getting the next child is 50-50%. Okay, अभी मैं लिख देती हूँ. Okay, that is XX, or we can say girl child is to XY, that is boy child, is how much? This is 1:1. Can we say that? This is 1:1. ठीक है. So कितना probability होता है बेटा? देखो, there is a family of father and there is a family of mother. ठीक है, मतलब इन दोनों को बन के एक family बन गया, and if they are crossing to get the child they're expecting, means there is a 50% of the possibility, and it is very expected also. ठीक है, either हमको 50% girl हो सकता है, 50% boy हो सकता है. ठीक है, so for this what we have to say here, we have to see in this. So this is 1, we know that, okay, the 50% of the chance, that is we are considering as the ratio 1. Okay, so now we have to understand 1 by what. Okay, so 1 by 2 is the correct option. So if you see here, they are saying, okay, so what they are saying here, so occurrence of 1 by 2, 1 by 5, 1 by 4. Okay, here 1 suggests what? 1 or, okay, so I will write here 50% is boy. Okay, it is the 1% or 50% is boy. Now, what does 2 indicate? So in 1 by 2, what does this 2 indicate? 2 indicate male and female. See, there are only 2 chances, right? So either the baby will be male or either it will be female. That is why 1 is the boy percentage, for 2 male or either female. Okay, so that is why it is 1 by 2. So please you have to keep a note of this. 1 by 2 kaisa aaya? 1 is for the 50%, that is 1 ratio of the boy, and the 2 is for male and female. Okay, so that is why answer to this question is very simple. That is option 1, 1 by 2. ठीक है, मतलब क्या है बेटा, कोई भी क्वेश्चन अगर आपको, if they are not asking son, if they ask you the girl child, then also the possibility is 50% only. Okay, then also the possibility is 50%. Either it is a boy, either it is a girl, it is always 1:1. That is 1:50%. Okay, is this clear to everyone? Yes. Then let's move on. Okay, then let's move forward. Okay, so now there is question 2n minus 1 minus 1 is known as. Okay, is known as. So what we have to see here, options are hypoaneuploidy, hyperaneuploidy, duplication, translocation. Okay, so now we have to understand the meaning of this terminologies to understand the term 2n minus 1 minus 1. Okay, so इसके लिए हम क्या करेंगे अभी? Now what we are going to do, we are going to understand the concept of mutation. So what actually is this? This is the concept of mutation. So here I have taken some PPT of the last class. मतलब मैंने पहले previous classes में ये part करवाया है mutation का. उसमें I had given very detailed explanation. So from there I have picked the PPT so that I will give you a clear understanding of this. Okay. So now we will look about the question point of view, but I'm giving this as a note. You can refer it later onwards. Okay. So mutation के दो types. There are 2 types of mutation. The first type is chromosomal mutation and the second type is gene mutation. Okay. So what actually is chromosomal mutation? Any change in structure or any change in number of chromosomes. ठीक है, क्या हो रहा है? Change in the structure or in change in the number of chromosomes. So both these come under the category of chromosomal mutation. Okay, and there's a second category called gene mutation. So what actually is gene mutation? There is a change or alteration in the chemical structure in the genetic level or molecular level. Okay, so that is alteration of chemical structure, that is gene mutation. Okay, so we have to talk about this now. We have to concentrate on the chromosomal mutation. So in chromosomal mutation, again there are 2 types: genomatic mutation and chromosomal aberrations. Okay, so genomatic mutation means what? Change in number of chromosomes. Okay, So here mutation is occurring by the change in number of chromosomes. Whereas chromosomal aberration में क्या हो रहा है? Change in structure of chromosome. The structure of chromosome में जो change हो रहा है उसको हम बोलते हैं chromosomal aberration. ठीक है? So now we have to continue with this. Okay, so we have to continue with the genomatic mutation. So here we are going to get many things. Okay. So in the genomatic mutation, there are two, categories, that is euploidy and aneuploidy. Okay, so you can read this later. Anyhow, you can read this later. Okay, but now we have to check the aneuploidy. Why? Because in our question they have asked two options: hyperaneuploidy and hypoaneuploidy. Right? So want to see? Let's go back. See, hypoaneuploidy and hyperaneuploidy. So that's why we are reading this slide of aneuploidy. Okay. तो यहाँ पे क्या हो रहा है? See, what actually is aneuploidy? Change in number of chromosome in a set. Is set. So what actually is a set? Set is nothing but the chromosome number. Okay, so consider this. This is one set and this is haploid set. That means this is the one set. ये set में एक ही chromosome होता है. That's why it is called as haploid. ठीक है? So इसमें देखो diploid. So what happens in the diploid? Here it is also a set, but here there are two chromosomes. That is why it is diploid. Okay? So if there is change here, okay, like अभी 2n के साथ एक और जुड़ गया. Okay, so 2n, there are already two chromosomes. Along with that chromosome, if there is another added in the same set, मतलब अगर वही set में another आ गया तो problematic है. That is called as aneuploid. Okay, so there is a limited set. For example, if something is diploid, in that diploid there are only two chromosomal set. Okay, if it is a haploid, there is only a one chromosomal set. तो वो होता है set. अभी उस set में कोई और एक chromosome add हो गया या delete हो गया, they have the different name for this. Now, hyperaneuploidy means addition of chromosome. Okay, addition of chromosome. If one is added, okay, there is a diploid set. In that diploid set, one more chromosome is added. Then we call it as trisomy. Okay, so this trisomy will give you the example of Down syndrome and Klinefelter syndrome. Okay, ये दो examples आपके NCERT में भी दिया है. That's why I have taken this only. Okay, I'm not specifying any different examples. I have taken examples from the NCERT itself. Okay, so 2n plus 1, this is trisomy. So similarly, in a set of diploid, I have added two chromosomes. That makes the tetrasomy. Okay, so this is addition of chromosome to the aneuploidy. Now here, hypoaneuploidy means deletion of chromosome. If I delete, okay, if I delete one chromosome from two diploid or two, chromosomal set or a diploid set, उसमें से अगर मैं एक chromosome निकाल दूँ, so then what happens is monosome. Okay, if I remove, okay, both the chromosomes from the diploid set, then there will be remain nothing. So hence the name nullisomy. Null means nothing. Okay, कुछ भी नहीं है. So that is null. So 2n minus 2 is actually nullisomy. So this is the question that is asked in your option. एक पूछा गया है hyperaneuploidy and another one asked is hypoaneuploidy. Okay, hyper means addition, hypo means deletion. Okay, so what you have understood by this? Now let's come here. If you see here, what you're going to write. So hyperaneuploidy means what? Here you are going to add. Okay, so add. Hypo means delete. Okay, delete. So here what is happening? 2n minus. So minus indicates what? Here something is getting deleted. Okay, something is getting deleted. Okay, so अभी दूसरे भी options हैं उसको देख के फिर we'll get back to the answer. हालांकि we can say that it is hypoaneuploidy, but still. Okay, now there are two more options called as duplication and translocation. So what actually is duplication? See beta, what happens is, okay, it is a segment or it is a set of chromosomes. Okay, there is one chromosome here. So in this chromosome, what happens is a segment of chromosome will occur twice. Okay, I have written here. This is one gene A, gene B, gene C, gene D, gene E, gene N, gene F, gene G. Okay, so these are the segment of chromosomes, or you can consider as gene or segments. So what happened here? This C segment is going to occur twice. Okay, so when it occurred twice, then what happened? A, B, C, D, then again there is a C. So how many times C has come? 2 times. So double the number. So this is called duplication. Okay, so duplication means what? A segment of chromosome in a chromosome will occur twice. Okay, now what is translocation? See, location means some place or some portal. Okay, trans means what? Transfer, transport. Okay, मतलब exchange करना. So here what is happening? Separation of chromosomal segment and its union to a non-homologous chromosome. What does that mean? See, this simply means that there are two chromosomes. ठीक है, अभी देखो बेटा. If I consider this is one chromosome and this is another chromosome, so these are all homologous. ठीक है, ये हो जाता है homologous. That means they are identical. If I write another chromosome here, and if I take these two as example, okay, if I take this with this, then it becomes the heterologous. Okay. Or wait, I'll write here non-homologous. The best way is non-homologous because that's what I have written here in the definitions. I will write this is homologous, this is non-homologous. ठीक है, तो अभी क्या हो रहा है? What does this mean? A segment of chromosome, okay, will separate and unite with the non-homologous chromosome. ठीक है, तो इसके भी दो टाइप्स हैं बेटा. The first type is simple translocation and the second type is reciprocal translocation. ठीक है, so in the simple translocation what will happen? You can see here. Okay. So you can see here, this is one chromosome and this is second chromosome. These are non-homologous. ठीक है, इसका सेट इसके अब एक सेट ऊपर है, इसका वाला नीचे वाले का सेट इसके नीचे है. But I have not mentioned it. Okay, you need not to worry. So here I have taken 1, 2, 3, 4, 5, 6, 7, one chromosome, then A, B, C, D, F, G, another chromosome. Now what will happen? This 6, 7 wala segment. So this part of segment will separate and bind with the non-homologous chromosome. Okay, so here I have shown you the result. What happens? So 6 and 7 का जो segment निकला था, it is going to join with the non-homologous chromosome. This is simple translocation. That means what? Somebody or a part of segment is transported from here to there. यहाँ से वहाँ गया है. बस ये होता है simple translocation. And another type of translocation is reciprocal translocation. There what happens? There is a reciprocal nature. मतलब part of segment is given from here to there, and again the similar kind of segment is received from there to here. Okay, exchange हो रहा है, reciprocal, reciprocate हो रहा है. Okay, so here you can see in the first chromosome I have written A, B, C, D, E, F, G, H. In the second I have written A, 1, 4, 1, 2, 3, 4, 5, 6. Okay, so here what happened? A segment of A, B, C and 1, 2, 3 are going to reciprocate. मतलब ये हो जाएगा उल्टा. मैं आपकी जगह में आप मेरी जगह में, like this. ठीक है, so here this is reciprocal. So this is reciprocal translocation. Okay, exchange. ठीक है, exchange हो जाता है. ये यहाँ पे जाएगा, फिर वहां से यहाँ को आएगा, like this. Okay, so now go back to the question. So here they are saying, okay, so hyponucleoid, hyperenucleoid. It is definitely not the hyperenucleoid because it is not going to be added. Something is created. But duplication means what? Duplication, repeated number of segments, which is wrong. Translocation means what? Exchange or translocation. A segment of chromosome is going to be locating, change its location. So this is also wrong. So the answer for this question is 1, hyper and 1. I hope this is clear. I have explained it in a much better way. Okay, बाकी वाले जो है आप पढ़ लेना. Okay, now next question is, during the application of DNA, opposite fragments are formed in the direction of 3 prime to 5 prime, 5 prime to 5 prime, 3 prime to 3 prime, 5 prime to 3 prime. Okay, so this is the question, and let's solve answering this. Now, what is replication? Replication means Okay, there is a DNA segment and we are making the identical copy of that DNA segment by utilizing the parent strand giving rise to the new daughter strands. Okay, so यहाँ पे in this picture you can clearly see that it is the parental strand, parental duplex. So here what is going to happen? The replication process has started to give the new strands. Okay, so this is new strand, this is new, and this is also new strand. Okay, so this is now. Now what will happen? So the ladder-like DNA fragment is going to separate first. So separation कैसे होगा with the help of enzyme? Okay, there is an enzyme called helicase. So that helicase will unwind and break the two DNA fragments or two DNA strands. So break हो जाने के बाद what happens? There will be called as SSB protein. They will bind here the separated DNA strands so that there should be stability. ठीक है, वो वापस से फिर जाके ज्वाइन ना कर जाए, इसलिए उनको अलग रखने के लिए यहाँ पे सिंगल स्ट्रैंडेड बाइंडिंग प्रोटीन लगाया जाता है. आफ्टर दैट देयर विल बी अ यूज ऑफ प्राइमर. ठीक है, सो देयर इज अ यूज ऑफ प्राइमर एंड आई थिंक ऑल ऑफ यू नो दैट प्राइमर ऑलवेज बाइंड्स टू द 3' रीजन. प्राइमर हमेशा 3' रीजन में जाके अटैच होगा. ठीक है, सो जहां पे 3' है वहां पे प्राइमर बनेगा. ठीक है, so now what happened? So here you can see there are two types of strands we are going to get. Okay, so from the replication, we are going to get the two strands. One is leading strand, also called as continuous strand, leading strand, or continuous strand. Okay, and the another one is okay, lagging strand. Another one is lagging strand or non-continuous strand or non-continuous strand. Okay, so these are the two types that we are going to get from the replication. Okay, so isme if you see here, this is the leading strand and this is the lagging strand. Leading strand will be moving or polymerization will happen from, okay, polymerization कैसे हो रहा है? Okay, so 5' to 3'. क्या हुआ देखो, प्राइमर जहां पे देखो प्राइमर कहाँ पे जाके अटैच होगा, प्राइमर वहां पे जाके अटैच होगा जहां पे 3' रीजन है. सो देयर इज अ 3' रीजन एंड दिस ग्रीन कलर इज द प्राइमर. ओके, सो प्राइमर 3' पे जाके अटैच हो गया एंड इट स्टार्टेड जनरेटिंग द न्यू डीएनए स्ट्रैंड. ठीक है, तो ये जो पॉलीमराइजेशन हो रहा है, this is happening from 5' to 3'. This is in the leading strand. Now comes to lagging strand. In the lagging strand, we are familiar with the opazygotic fragments. मतलब it is not continuous. ये part में होता है. ठीक है, formation of strand. So here what happened? So again this is 3'. ठीक है, तो जहाँ पे 3' होगा वहाँ पे 5' होगा opposite में. So 5' to 3'. Again 5' to 3'. So like this. Okay. So even in the leading strand, the polymerization will happen from 5' to 3'. Even in the lagging strand, ये आपको याद रखना है, from 5' to 3', even in the lagging strand. ठीक है? So this is the overcyclic fragment. Now let us see here. Okay. So during the replication of DNA, Okazaki fragments are formed in the direction of 5' to 3'. तो मतलब उसी डायरेक्शन में पॉलीमराइजेशन हो रहा है, तो उसी डायरेक्शन में हम लोगों को मिलेगा ना Okazaki fragment. So it is as simple. Okay, now next question. Okay. So I'll read this question for you. The given figure is a schematic breakup of the phases or stages of cell cycle. Select the correct option. Okay, so this is the diagram. Please have a look at this diagram. And here options are: A represents division of cytoplasm, B is telophase, which is just reverse of prophase, C is the best phase to count total number of chromosomes in any species. In D stage, replication of DNA takes place. Okay, so this is the options. Now, let us understand the question and the cycle. So here the question is asking about the cell cycle, and the question is taken from the chapter cell cycle and cell division. Okay, तो यहाँ पे मैंने एक डायग्राम दिया है for your better understanding. This is cell cycle. Okay, so in the cell cycle, what happens? Firstly, there are two phases. Okay, so first में क्या होता है? There is an interphase and there is M phase, right? So what happened in interphase? There are further phases G1, S and G2 phase. Okay, so in M phase, what we are going to see in M phase, again there are two categories, that is karyokinesis and okay, one is karyokinesis and another one is cytokinesis. ठीक है, ये कहाँ से आ रहा है? M phase से. ठीक है, it is coming from the M phase. ठीक है, M phase में दो है phases, that is karyokinesis and cytokinesis. Now, what is karyokinesis? Here, division of nucleus will occur. Okay, so division of nucleus, or we can say the nuclear matter. So here, division of cell. Okay, so the division of cytoplasm. So here we can say division of cytoplasm. So first क्या होगा? First karyokinesis होगा, उसके बाद cytokinesis होगा. So which are the stages in karyokinesis? Again, there are stages. The stages are T, mat. ठीक है, प्रोफेज, मेटाफेज, एनाफेज, टेलोफेज. ठीक है, ये पांच हो गए. सॉरी, ये चार हो गए. So now we can see here. ठीक है, तो यहाँ पे you can see here cells growth is going to start from the interface. So ठीक है, ये जो part है बीच में, the blue color one is interface. Okay, this is interface and this is M phase. This much is M phase. In the interface, there is G1, S and G2. So G1 phase में क्या होता है? The cell will grow. Okay, double its cytoplasm, double its contents. उसके बाद क्या होता है? S phase में DNA replication will happen. ठीक है, अभी देखो साइटोप्लाज्म सब डबल हो गया, सेल ऑर्गेनल्स डबल हो गया. अभी न्यूक्लियस इज द मेन पार्ट एंड द जेनेटिक मटेरियल इज वेरी इंपॉर्टेंट. सो दैट्स व्हाई वी हैव टू रेप्लिकेट द डीएनए आल्सो. ठीक है, जब डीएनए भी दो में होंगे, देन वी कैन इक्वली सेपरेट among the genetic material among the two daughter cells, right? That is why DNA replication. Then what happens for the preparation of mitosis or meiosis? That is in the G2 stage. So then starts the mitotic phase or mitotic division or meiotic division. So here what happens? Mitosis will occur in these stages. Okay, so first it starts with prophase, anaphase, metaphase, anaphase and telophase. So cytokinesis is the last, and then again we enter into the cell cycle. Okay, so this is what. ठीक है, तो अभी क्या होता है? प्रोफेज में what happens? You have already know this, you have studied all this. I'm not going to repeat it. ठीक है, मेरे बच्चे बहुत चालाक हैं, आप एक बार जाके रिवाइज कर लेना. नाउ लेट अस गो टू द ऑप्शन. हम लोग यहाँ पे ऑप्शन देखेंगे. चलो, A is what? A is actually the mitosis. Mitosis. So mitosis is what? Mitosis is division of nucleus, right? So A is wrong. Okay, A is wrong. It doesn't represent the division of cytoplasm. So which represents the division of cytoplasm? Cytokinesis, that is C here. Okay, now B. So B is telophase, which is just the reverse of prophase. Telophase is reverse of prophase, but B is not telophase. B is anaphase. ठीक है, B क्या है? B होगा anaphase. ठीक है, so this is also wrong. Then C. C is the best phase to count total number of chromosomes in any species. No, no, वो मेटाफेज है. ठीक है, so यहाँ पे जो C लिखा है, C जो है ना बेटा, ये क्या होता है? डिवीजन ऑफ साइटोप्लाज्म. ओके, डिवीजन ऑफ साइटोप्लाज्म होता है यहाँ पे. ओके, बट व्हाट दे आर सेइंग हियर, ओके, इसको मैं हटा दूं तो द बेस्ट प्लेस टू द काउंट द क्रोमोसोम नंबर इज मेटाफेज. दैट इज मेटाफेज. ओके, बहुत अच्छे स्टडी भी कर सकते हैं और काउंट करने के लिए भी मेटाफेज भी बहुत अप्रोप्रिएट है. सो space, what happened? The replication of DNA takes place. So 4 is the answer for this question. Are we clear with this? Very easy question. Okay. So now select out correct combination about incomplete dominance. ठीक है, incomplete dominance के बारे में आपको options choose करना है. So this is the table. Please have a clear look at the table. ठीक है, एक बार table को अच्छे से देखो, then we'll start answering. Meanwhile, please remember what is incomplete dominance. Okay, incomplete dominance क्या है आपके लिए? Incomplete dominance is actually not given by Mendel. It is non-Mendelian inheritance होता है. ठीक है, तो अगर मैं कहूँ R R और RR है, अगर रेड और व्हाइट को हम क्रॉस करवाएं तो हमको क्या मिलेगा? कैपिटल R स्मॉल r. सो इसको रेड होना चाहिए था, बट इट इज पिंक. दिस इज इनकम्प्लीट डोमिनेंस. मतलब ये जो रेड डोमिनेंट है, इट इज नॉट कंप्लीटली डोमिनेटिंग ओवर व्हाइट. ठीक है, तो ये होता है पिंक. सो दिस इज एन एग्जांपल दैट आई हैव टू टेल यू अबाउट द इनकम्प्लीट dominance. So इसको हम बोलते हैं incomplete dominance, and this is taken from your NCERT only. ठीक है, ये NCERT का ही statement है. ठीक है, तो ये होता है thing. Now you can analyze in this way also. ठीक है, इसी तरीके से आपको सोचना है. ठीक है, now अभी यहाँ पे हम लोग क्या करेंगे? What we are going to do? See, it's very simple. We have to take capital A small a, capital B small b. ठीक है, so now we'll start to cross among them. ठीक है, हम लोग अगर क्रॉस करेंगे ना, ठीक है, then capital A, capital B, capital A small b, small a capital B, small a small b. So what are these things? These are the gametes. ठीक है, तो ये क्या हो गया? We can consider this as the parent. इसको हम ले लेते हैं parent. So if we are crossing among these, what are we get? We get these gametes. Okay, so these are the gametes. ठीक है, ये gametes है. Now when we cross them, okay, when we cross them, what are we going to get? ठीक है, इसको हम अगर क्रॉस करेंगे तो हम क्या मिलेगा? तो ये हमको देखना है. सो लेट मी राइट हियर. ओके, एक मिनट बेटा, just give me a second. Okay, so you can see here. Okay, I have taken the same picture because same kind of question was given in the first question also, if you remember. So instead of writing, I have cut short my time. So here you can see these are the gametes. Okay, so among them we will consider one as male and another one as female. So gametes तो same है ना बेटा, देख लो same gametes. यहाँ पे यहाँ 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 फिर यहाँ यहाँ. ठीक है, तो ये same gametes. ठीक है, now what actually the question is asking? Okay, so देखो they are asking about the genotype. Okay, genotype एक ही है. Okay, 4 options have the same genotype, that is capital A small a, capital B small b. ठीक है, so now first we have to find how many gametes we are going to get. How many gametes we got? Okay, so sorry, I will write here. Okay, so number of gametes we got or 4. ठीक है, 4 मिल गए. तो here what we can do, यहाँ पे हम क्या कर सकते हैं? इन दोनों को कैंसिल कर दो. क्यों? क्योंकि यहाँ पे दिया है 4 टाइप ऑफ गैमेट्स, तो इन दोनों में एक कैरेक्टर है. नाउ व्हाट वी हैव टू लुक, वी हैव टू लुक द फिनोटाइप. ठीक है, फिनोटाइप देखना है, तो उस फिनोटाइप में आइदर 9 फिनोटाइप है या 4 फिनोटाइप. ठीक है, दैट वी हैव टू लुक. So that we will look here in this, okay, in this book or in this diagram. तो यहाँ पे हम लोग देखेंगे बेटा. See how we are going to look. I think all of you know that. Okay, every gene has 2 alleles. One is dominant and another one is recessive. ठीक है, अगर डोमिनेंट है, if there is dominant, then the recessive allele will not show itself. Okay, so अगर dominant है तो dominant ही express होगा. We have to remember that. Okay, now so consider the first letter first. ठीक है, so A capital, B capital. Okay, so this is both dominant. This is also same. A capital, B capital. First वाला A capital, B capital, A capital, B capital. ये same है, same type of phenotype. Same phenotype, same phenotype, same phenotype, same phenotype, same phenotype. ठीक है, जो मैंने रेड में लिखे हैं, रेड में लिखे हैं, हियर व्हाट यू हैव टू राइट, दीज आर ऑल सेम फिनोटाइप्स. ओके, सो सेम फिनोटाइप्स है रेड वन. ठीक है, सो 1 2 3 4 5 6 7 8 9, तो टोटल कितने हो गए? 9 नंबर. नाउ आई विल चेंज द कलर. नाउ आई विल चेंज द कलर. आई विल टेक ब्लू. ठीक है, अभी देख लो, कैपिटल ए स्मॉल बी. ठीक है, एक कैपिटल रहेगा, एक स्मॉल रहेगा. ठीक है, तो ए को कैपिटल लेते हैं, बी को स्मॉल लेते हैं. देखो, ए कैपिटल है, बी स्मॉल है. ए कैपिटल, बी स्मॉल, और यहाँ पे ए कैपिटल, बी स्मॉल. ठीक है, अब ब्लू से हम क्या करेंगे, ये भी अगेन सेम फिनोटाइप है. ठीक है, सो अगेन हियर आल्सो वी आर गोइंग टू राइट. ठीक है, सेम फिनोटाइप. जिस फिनोटाइप इस कितने हो गए यहाँ पे? 3. ठीक है, 3 ऑफस्प्रिंग्स हैं. अगेन आई विल टेक द डिफरेंट कलर. आई विल टेक द डिफरेंट कलर ग्रीन. ठीक है, यहाँ पे ले लो, स्मॉल ए कैपिटल बी, स्मॉल ए कैपिटल बी, स्मॉल ए कैपिटल बी. तो ग्रीन में भी क्या हो गए? सेम टाइप ऑफ फिनोटाइप, सेम अनदर टाइप ऑफ फिनोटाइप. तो यहाँ पे भी कितने हैं? अगेन 3 नंबर. अगेन आई विल टेक द डिफरेंट कलर. एक ही बचा है, अभी तो एक ही बचा है. लेट मी टेक व्हाइट. सो दिस. ठीक है, जो व्हाइट में लिखा है ये भी सेम है, ये एक ही है, दैट इज कैपिटल स्मॉल ए स्मॉल बी. ओके, ये एक ही है. तो टोटल कितने फिनोटाइप्स हो गए? यू टेल मी, टोटल हाउ मेनी फिनोटाइप्स दैट यू कैन काउंट हियर? यू कैन काउंट 1 2 3 4. Total there are 4 phenotypes. I will write here number of phenotype is again 4. ठीक है, तो एक ही phenotypes के डिफरेंट डिफरेंट जीनोटाइप है बेटा. एक ही phenotype के डिफरेंट डिफरेंट जीनोटाइप है. ठीक है, बट phenotype is same. ठीक है, कितने phenotype है? 4. Now let's get back here. So what are the options that we have? This is the genotype. Okay, the 4 type of gametes. Okay, both are correct. Now 9 phenotype, no. The 4 phenotype. 4 is the answer for this question. Is everybody clear with this? ये clear हो गया सबको? चलो, बहुत अच्छी बात. Now let's go ahead. During the process of pro-insulin into the mature insulin, the options are C-peptide is added to pro-insulin, C-peptide is removed from pro-insulin, B-peptide is added to pro-insulin, B-peptide is removed from pro-insulin. ठीक है, तो ये हमारे ऑप्शन हैं. So for that, let's go and see the next slide. ठीक है, इस स्लाइड में हम देखते हैं बेटा क्या दिया गया है. ठीक है, so this first one. Okay, so this is the pro-insulin. Okay, this is pro-insulin. ठीक है, ये क्या है? pro-insulin है. And this is insulin. Okay, ये जो यहाँ पे लिखा है ये insulin है. Now here in the pro-insulin you can see the 3 protein chains or amino acid chain, that is A chain, B chain and C. Where is C? This is C chain, C peptide. So here what happened in the pro-insulin, there is A plus B plus C. All the 3 is there in the pro-insulin. ठीक है, अभी इंसुलिन में क्या होता है? फिर इंसुलिन में what do you see in the insulin? In the insulin you can see only A plus B. You can see only A plus B but minus C. That means there is a removal of C. ठीक है, so what is the verdict of this? What we can say? Okay, hence removal of C peptide, removal of C peptide. देखो यहाँ पे C peptide निकल गया. So this is A, this is B. Now this is called as biologically active insulin. ठीक है, this is very easy. तो यहाँ पे क्या है, हमको लिखना है प्रोसेसिंग ऑफ प्रोइंसुलिन टू इंसुलिन में क्या होता है. C peptide is added. No. C peptide is removed. Yes. B peptide is added. No. B peptide. No. Okay. So 2 is the answer for this question. Clear है सबको? 2 is the answer for this question. ठीक है. चलो. Now choose the correct pattern of fluid for counter current in vasa recta. ठीक है. vasa recta की counter current में क्या होता है उसका यहाँ पे pattern देखना है. So for this, let us understand what actually is counter current mechanism. So this counter current mechanism you are going to study in urine formation. ठीक है, जो यहाँ पे हम लोग excretion के बारे में पढ़ते हैं, formation urine के बारे में पढ़ते हैं, वहाँ पे we are going to understand this. So here what happened? Okay, here you are going to see. ठीक है, यहाँ पे पहले हम क्या लेंगे, जो अंदर का ट्यूब है that is vasa recta. ठीक है, जो बाहर वाला ट्यूब है, okay, so this is loop of Henle. Okay, so this is loop of Henle. ठीक है, तो ये loop of Henle है. Now what is happening? So loop of Henle में कौन सा है? This is ascending. Okay, so this is ascending loop and this is descending loop of Henle. ठीक है, तो ये कैसे हो रहा है, किस डायरेक्शन में जा रहा है? ऐसे. वेट एक मिनट, ऐसे नहीं, एक मिनट मैंने उल्टा लिख दिया यहाँ पे, मैं इसको क्लियर कर लेती हूँ. ऐसे लिख देती हूँ. ठीक है, ये होता है डिसेंडिंग एंड ये होता है असेंडिंग, है ना, क्योंकि ऐसे ही है. सो फ्रॉम द डिसेंडिंग व्हाट हैपेंड, डिसेंडिंग मींस गोइंग डाउन. ठीक है, तो नीचे जा रहा है, नीचे जाते जाते ऐसे ऊपर जा रहा है, दिस इज असेंडिंग. सो इन दिस डायरेक्शन इन द लूप ऑफ हेनले फिल्ट्रेट इज मूविंग. ओके, मतलब जो अल्ट्रा फिल्ट्रेशन और ग्लोमेरुलर फिल्ट्रेशन हुआ था इन द यूरिन फॉर्मेशन, उसके बाद क्या होता है बेटा, द फिल्ट्रेट जो बना है प्रेजेंट इन द Bowman's capsule will come to PCT. From PCT, it will come to the descending limb of loop of Henle. Then it will come to loop of Henle and then ascending limb of loop of Henle. So it is in this direction. ठीक है, वहां से फिर DCT को जाएगा, DCT से collecting duct को जाएगा. Now, so there are small, thin blood capillaries which are surrounding the loop of Henle. We call them as vasa recta. ठीक है, हम उनको बोलते हैं vasa recta. So this vasa recta is in the opposite direction. मतलब यहाँ पे डिसेंडिंग वैसा रेक्टा है और यहाँ पे असेंडिंग वैसा रेक्टा है. दैट मीन्स व्हाट, डिसेंडिंग मतलब ब्लड कैपिलरी में ब्लड ऐसे जाएगा. ठीक है, सो लाइक दिस इट विल गो. सो दिस मीन्स व्हाट, ओके, सो फिल्ट्रेट इज मूविंग इन वन डायरेक्शन एंड द ब्लड इज मूविंग इन अनदर डायरेक्शन. This we call it as counter current. Counter means opposite, current means flow. Okay, mechanism is that is what happening here. ठीक है, तो एक इस डायरेक्शन में एक इस डायरेक्शन में, दिस इज काउंटर करंट मैकेनिज्म. ठीक है, नाउ द क्वेश्चन वाज आस्किंग अबाउट द वैसा रेक्टा. ठीक है, तो वैसा रेक्टा में क्या हो रहा है, वैसा रेक्टा में क्या हो रहा है, राइट साइड में डिसेंडिंग है जो नीचे जा रहा है, डिसेंडिंग में नीचे, असेंडिंग में ऊपर. ठीक है, ये हमको अभी यहाँ पे देखना है. ठीक है, डिसेंडिंग में नीचे तो आ रहा है, असेंडिंग में भी नीचे, नो. डिसेंडिंग में नीचे आना चाहिए, ऊपर जा रहा है, नो. डिसेंडिंग में नीचे आना था, ये तो ऊपर जा रहा है, नो. डिसेंडिंग में नीचे, असेंडिंग में ऊपर. 4 इज द आंसर फॉर दिस क्वेश्चन. सी, काउंटर करंट मैकेनिज्म में ऑस्मोलैरिटी आता है, ठीक है, 300 से 1200 तक जाता है, फिर वापस जाता है, वहां पे हाइपोऑस्मोलैरिटी, हाइपर ऑस्मोलैरिटी सब आता है. That is counter current mechanism. I am not explaining that mechanism here. Just you have to look at the pattern. मतलब जो फिल्ट्रेट किस फ्लो में, किस डायरेक्शन में फ्लो कर रहा है, एंड ब्लड कैपिलरी में ब्लड किस डायरेक्शन में फ्लो कर रहा है, एंड ऑलवेज फ्लो इन द ऑपोजिट डायरेक्शन, हेंस द नेम काउंटर करंट मैकेनिज्म. एंड हियर दे आर आस्किंग द वैसा रेक्टा. वैसा रेक्टा में डिसेंडिंग लिम्ब से आके असेंडिंग लिम्ब में जाएगा. ठीक है, सो डी इज द आंसर. नाउ द नेक्स्ट क्वेश्चन, स्पेस बिटवीन the two adjacent neurons where the chemical transmitter is released known as, okay, synaptic vesicle, electrical synapse, synaptic cleft, terminal button. ठीक है, तो ये सारे ऑप्शंस हैं. So now let us understand, this question is taken from the chapter neural control and coordination. ये बहुत important concept है ये. So here what they're saying is देयर आर टू एक्सॉन टर्मिनल, ठीक है, वहां पे एक्सॉन नॉब होता है, तो वो जो नॉब्स हैं दो, उनके बीच में कुछ न्यूरोट्रांसमीटर पास होते हैं, जो केमिकल ट्रांसमीटर्स, तो वो प्लेस का नाम है क्या, उस स्पेस का क्या नाम है, वो दैट वी हैव टू सी इन दिस डायग्राम। यू कैन से दैट very clearly. This is one neuron and another neuron. So here what is happening here, there is a communication occurring between those two neurons and that communication is highlighted here. उसको हम लोगों ने इसको बड़ा करके यहाँ पे दिखाया, हाईलाइट, ठीक है, और इसको हाईलाइट कर रहे हैं हम इन दिस डायग्राम, ठीक है। हियर व्हाट हैपन, दिस इज एक्सॉन टर्मिनल, दिस इज वन एक्सॉन टर्मिनल एंड दिस इज एन अनदर न्यूरॉन, ठीक है, दिस इज न्यूरॉन 1 2, दिस इज न्यूरॉन 1, ठीक है, तो यहाँ पे हो रहा है, ठीक है। So here what is happening, so action potential is came here, action potential will be coming here, so action potential will bring the what signal, so this signal will make these vesicles, okay, this synaptic vesicles, these are the vesicles, okay, so these vesicles containing neurotransmitters will start to move towards the base of the knob, synaptic knob, ठीक है, ये जो knob है इस knob के base में आएगा। So when it comes to the base, what happens? It will release all these neurotransmitters. So these small pink color balls are neurotransmitters. These are released. Okay. Then these neurotransmitters are accepted by the receptors which are present in the second neuron. Okay. यहाँ पे neurotransmitters हैं. These neurotransmitters are accepted by these receptors, and this receptor site is the receiving of another neuron. तो यहाँ से क्या होगा, सो दिस एक्शन पोटेंशियल इज पास्ड, ठीक है, एक्शन पोटेंशियल दूसरे न्यूरॉन को पास, ठीक है। नाउ दिस गैप इज व्हाट दे आर आस्किंग, दिस गैप इज सिनेप्टिक गैप और आल्सो कॉल्ड एज सिनेप्टिक लेफ्ट, synaptic left or synaptic gap. So this is what you need to remember. This is what they have asked. Okay. तो यही पूछा गया है यहाँ पे. So the question is space between two adjoining neurons where the chemical transmitter is released. Not a vesicle तो नहीं, देखो ऐसा था ना vesicle उसके अंदर neurotransmitter. This is not the thing. Electrical impulse means what? It is action potential. ये हो गया आपका action potential. Even this is also not. ये भी नहीं, ये भी नहीं. Terminal but is what the knob. Okay, so that synaptic knob. Okay, that synaptic knob or the axon terminal. Over there. So answer to this question is C, synaptic cleft. Synaptic cleft is the answer. Okay. Okay. So now there's another question on the screen, and this is the last question for the day. Okay, because we are in the end. So I'll read out the question for you. Read the following four statements A, B, C, D, and select the right option having both correct statements. मतलब you have to find the two correct statements among these four statements. Okay, this is okay. First statement A is Z scheme of light reaction takes place in presence of PS1 only. Only PS1 is functional in cyclic photophosphorylation. Cyclic photophosphorylation results in synthesis of ATP and NADPH. Stromal lamellae lack PS2 as well as NADP. Okay, so these are the statements, and now you have to answer them. अभी इसका हम लोग को answer करना, okay? Okay, so now I will write here. मतलब I will write like a flow. तो मैं यहाँ पे लिखूंगी. So till now I will summarize what I have understood about photosynthesis. ठीक है? So क्या हो रहा है? In the process of photosynthesis, there are mainly two phases. है ना? There are mainly two phases. Which is the first phase? The first phase is light reaction. Light reaction, and the second one is dark reaction. ठीक है, तो एक हो गया लाइट रिएक्शन और एक हो गया डार्क रिएक्शन. ये फोटोसिंथेसिस के दो मेजर फेजेस हैं. ठीक है, तो लाइट रिएक्शन में क्या होता है? अगेन देयर विल बी नॉन साइक्लिक फोटोफॉस्फोरिलेशन एंड साइक्लिक फोटोफॉस्फोरिलेशन. ठीक है, So these two parts will come under light reaction. So here non-cyclic is also called as Z scheme. Okay, non-cyclic को हम क्या बोलते हैं? Z scheme बोलते हैं. So in dark reaction again we are going to see the two categories, that is C3 plants and C4 plants. Okay, C3 plants are the most of the plants which you are going to see on the land. जो आप लैंड में देखते हो ना वो होते हैं C3 plants. तो ये C3 plants में normal plants है, normal mesophytic plants. But C4 plants are very efficient, very adapted plants. For example, for C3 plant, there is 10 liter of water needed. Okay, to absorb within 5 hours. ठीक है, 5 hours में कंसिडर करो ये 10 लीटर वाटर कंज्यूम करता है. ठीक है, तो हमारा C4 साइकिल जो है ना ओनली 5 लीटर एब्जॉर्ब करता है. ओनली 5 लीटर इज नीडेड. आई एम नॉट टॉकिंग अबाउट जस्ट एब्जॉर्प्शन, आई एम टॉकिंग अबाउट द नीड ऑफ द प्लांट. इसको 5 लीटर की नहीं है, मतलब 5 घंटे में ये 5 लीटर पानी देता है. दैट इज सफिशिएंट. दैट मींस दीज आर मोर इफेक्टिव. ओके, C4 आर मोर इफेक्टिव. प्लीज रिमेंबर दैट. Okay. Okay. Now C3 cycle जो डार्क रिएक्शन में होता है. Okay, so डार्क रिएक्शन में जो C3 साइकिल होता है उसको हम नेम देते हैं केल्विन साइकिल. Okay, there will be a Kelvin cycle here. ठीक है, C4 में होता है Kranz anatomy. Okay, Kranz anatomy तो वो होता है एक. ठीक है, so these two are coming under dark reaction. Now what do we expect as the result in non-cyclic photophosphorylation? From the non-cyclic photophosphorylation we will get oxygen plus ATP plus NADPH. ये सब कुछ हमको मिलता है नॉन साइक्लिक फोटोफॉस्फोरेशन में. साइक्लिक में हमको सिर्फ ATP मिलता है. ATP only, we will get only ATP. Did you understand my point? Okay, so here what is going to happen in the process of photosynthesis? There are two main phases, light and dark reaction. Light reaction, non-cyclic photophosphorylation, cyclic photophosphorylation will occur. तो नॉन साइक्लिक में कौन कौन इन्वॉल्व होते हैं? बोथ PS1 एंड PS2 दोनों इन्वॉल्व होते हैं. बोथ PS1 and PS2. In the cyclic only PS1, only PS1 here both. ठीक है, तो नॉन साइक्लिक में we are going to get oxygen, ATP and NADPH, whereas in the cyclic only ATP will be originated. So this ATP and NADP are used in the dark reaction. ठीक है, so this is one step. Now अभी हम लोग पीछे जाते हैं. I will tell you one more thing. ठीक है, मैं आपको कुछ और बताती हूँ. देखो बेटा, there is grana, है ना? In the chloroplast you're going to see the grana. So in that grana you're going to see that pile of coin. That each coin is called as thylakoid, है ना? each को हम बोलते हैं thylakoid. इसको हम बोलते हैं thylakoid. इसको together हम बोलते हैं grana and इसको हम बोलते हैं stroma lamina. So inside these there will be presence of quantasomes or photosystem. ठीक है, photosystem होते हैं इन सारे के अंदर. इन सारों के अंदर photosystem होता है. ठीक है, so ये सारे. So here I will write PS1 है जो मैंने white में लिखा है. ठीक है, so PS2 मैं लिखती हूँ ब्लू में. ये PS2 हमेशा एक के ऊपर एक, मतलब सब क्या कहते हैं, एक के ऊपर वो ओवरलैप हो जाते हैं. ठीक है, एक्सपोज नहीं होते डायरेक्टली टू द सन, बट फिर भी क्या करते हैं, ये भी काम करते हैं. दे विल आल्सो एब्सॉर्ब द केमिकल एनर्जी, दैट इज लाइट एनर्जी कन्वर्टेड इनटू द केमिकल एनर्जी, वो भी करते हैं. ठीक है, लेकिन PS1 जो है वो बहुत फैला हुआ होता है. दैट विल बी प्रेजेंट इन द स्ट्रोमा लैमेला एंड इन द सराउंडिंग्स ऑफ द थाइलाकॉइड्स. ठीक है, तो ये आपको याद रखना है. ठीक है, और सो हम लोगों ने जो पढ़ा है, ओके, नॉन साइक्लिक एंड साइक्लिक फोटोफॉस्फोरेशन दैट विल हैपन इन द लुमेन ऑफ द थाइलाकॉइड. ठीक है, तो इस लुमेन ऑफ द थाइलाकॉइड में यहाँ पे एक एंजाइम कॉम्प्लेक्स होता है. ठीक है, विच इज कॉल्ड एज FNR, ओके, पैराडॉक्सिन इनहिबिटरी रिडक्टेज एंजाइम. ये होता है अटैच्ड टू लुमेन फेसिंग टुवर्ड्स स्ट्रोमा. बाहर का पूरा स्ट्रोमा है, इमेजिन करो ये सारा स्ट्रोमा. ठीक है, ये सारा स्ट्रोमा, ये सारा स्ट्रोमा. Because FNR is facing towards stoma. FNR किधर face कर रहा है? Stoma. Now let us read all the sentences once again. Okay. Okay. That scheme of light reaction takes place in presence of only PS1? No. क्यों? मैंने आपको बोला था यहाँ पे देखो Z scheme में PS1 and PS2 दोनों हैं. ठीक है? So that is why first statement is wrong. Okay. Now only PS1 is functional in cyclic photophosphorylation. That is true. Here what you can see in the cyclic photophosphorylation only PS1 is involved. This is correct statement. ये जो statement है B statement, this is correct statement. Okay, now see. Cyclic photophosphorylation results in synthesis of ATP and NADPH? No. No. It results only in ATP, सिर्फ ATP. Okay, so hence this is also the wrong option. D. Stromal lamellae lack PS2 as well as NADP reductase. That is definitely correct. Why? Because you can see here, this is stromal lamellae, right? Here PS1 is there, not PS2. Whereas FNR is also mounted. FNR is NADP reductase, redoxin NADP reductase. So D is the correct statement. Okay. So B and D are correct. Hence the answer for this question is 4. Okay. So these are all the questions that I have taken. So if you have any further questions, you can ask me. अगर आपको कुछ पूछना है तो आप पूछ लीजिए. Okay. If there is nothing, then I will launch the NPS poll. Please make sure you give me the feedback for my better improvement. Okay. So मैंने poll launch कर लिया है. You can give your opinion in the poll. If you have any doubts, you can ask the doubts. So this is your homework. You have to solve the remaining questions from the. Okay, so हाँ PPT में तो मैंने क्वेश्चन नहीं दिया है, but जो आपकी क्वेश्चन पेपर है, ठीक है, from the question paper you have to solve from the question paper. आपको सारे क्वेश्चन सॉल्व करना है क्वेश्चन पेपर के, then you have to solve more and more model questions and previous year questions. This is your homework, and thank you. Okay, so if there is nothing, then we will end the meeting. Yeah, अगर कुछ नहीं है तो हम end.